Welcome back. On this video, we will continue our study of the visual system. So, on our last video, we already covered a few of the important structures using this diagram. Let's continue looking at a few more parts. And our focus will be on the inner structures, starting with the retina. Really, retina can be considered as an outgrowth part of the brain. See, the optic nerve, which we mentioned earlier, is outpocketing of the brain and then continues as this layer which we call retina. And this structure contains millions of photoreceptor cells that respond to light energy, transferring it into nerve impulses that then travel down to the brain's primary visual processing area and for further analyzing. And one thing to note here is that this structure is two-layered. We have an enlarged view of this area, and this is where we can see the two layers especially well. So we have outer pigmented layer and the inner layer of neurons. Let's look at the pigmented layer a little bit. So this is a single cell thick layer and functions to absorb the light to prevent it from scattering, and stores A vitamin, as well as helps with the phagocytosis of the photoreceptor cell fragments. Neural layer instead is the business end, if you wish. This is composed of three types of neurons. Photoreceptors, which then pass the messages to bipolar cells, which then allow messages to travel further to the ganglion cells. And it is the ganglion cells that exit the eye as an optic nerve. And another important structure to notice here is the optic disc. You might have heard about this as the blind spot. This name is because the area lacks photoreceptors, so we cannot see what falls on these regions. What I would like to do next is to focus on the neural layer. And here, in particular, the two types of photoreceptors, rods and cones. Rods respond to the dim light, being more focused on the shapes and sharpness of the image rather than the color information. And in fact, they are more numerous on the peripheral areas of the visual field. And here is a fun fact. If you wish to see something on a low light level conditions, like a star, try having a look a little past it so that the light falls on the peripheral area of your retina where you have more rods, and you will be able to see it better. And then of course are cones, which are optimally functioning at the bright level light and are responsible for the color vision. They are more numerous in the center of the field of view. And what I want to show here is a picture of real neural layer of the retina. See how it looks like our diagram. And there is one more important thing I want to mention here, and it is the fluids inside the eye. As we saw earlier, it was the lens and the suspensory ligaments that we can use to separate the anterior and the posterior segments of the eye. And within the posterior segment, the fluid is known as vitreous humor. This transmits the light and supports the posterior surface of the lens, as well as holding the neural layer on its place. And to do this, we must have a right amount of pressure there. Not too much, but not too little. And then on the anterior segment, we have aqueous humor, which, unlike the vitreous humor of the posterior segment, is constantly being regenerated. And here we are, back to this diagram. Now I feel that we have done a great job of covering the anatomy of an eye, and we should move our attention to the physiology involved. So let's do just that. So to start with, I think we should remind ourselves of the basics of electromagnetic spectrum. And as we can see on this diagram, the wavelength can differ a lot, from the scale of the buildings or even skyscrapers, as is the case with the radio waves, to the very small waves that are the size of subatomic particles, known as gamma rays. And the visible light that we are interested in falls somewhere in between these. And the visible light, within there are different colors that depend on the wavelength of the electromagnetic radiation. And different photoreceptors are differently sensitive to different colors. The other concept that we need to be aware is the fact that light bends. And this is known as refraction. This happens when the light passes from one transparent media to another transparent media, and the path is at an oblique angle. A good example would be from liquid to air, as we see here. And this is where the lens of an eye becomes important. The curvature can be used to bend the light. And it is now just a matter of how the lens is curved in terms of how the light gets bent. And you might also notice how this is also used as the concept behind the eyeglasses. And in an eye, the lens is able to adjust its curvature to allow for fine focusing. So we can move from distant vision to close vision, usually without any problem and quite fast too. But eyes are actually adapted ideally to distant vision. And for close vision, we must adjust the lens, constrict the pupil, and we may even slightly change the overall shape of an eyeball. So, how does the phototransduction work on a neural cell level? Well, photoreceptors' outer segment is what receives the light and contains visual pigments that react to light by physically changing their shape. And then the inner segment then joins to the cell body. And then the cell body is connected to the synaptic terminal from where the messages get passed onwards as another electrical impulse, an action potential. And actually what we end up finding is that our photoreceptors are very vulnerable to light, and therefore they need to be renewed almost every 24 hours. This is why we have so much phagocytosis going on here in the eye. It makes sense now, doesn't it? You can study the process of phototransduction in a greater detail from your textbook. What I want us to focus for the rest of this video instead is the processing of visual information in the brain. So in our earlier discussion, we got to the level of the ganglionic cells. The information has traveled from photoreceptors to there. And from here, the information travels to the optic nerve, which leaves the eye. And actually, information from each eye travels down the optic nerve until it crosses over at the structure known as optic chiasm. We saw it in our sheep brain dissection video. And then optic tracts continue to the thalamus, and from there onwards to the primary visual cortex in the occipital lobe. And this is where we first have a conscious perception of a visual image. And actually, another interesting thing here is the fact that because both eyes view the same image from a slightly different angle and visual cortex fuses these images, we get a three-dimensional image, allowing us to have a sense of depth perception. For example, people using only one eye would not be able to perceive depth like this. And this is where our videos on the visual system will end. I would encourage you to study the other systems more independently.